Okay, welcome to your Kin 225 class for Monday, March 23rd. As you can see from the PowerPoint, we're going to review carbohydrate metabolism, and I've got some enzyme questions. And then the main topic of today's class is whole body glucose metabolism. So a review question for you here. Which pathway produces the most ATP? The correct answer here is D, the electron transport chain. So if you look at all these other pathways, glycogenolysis, glycolysis, Krebs cycle, you can see from this slide, glycogenolysis is essentially the breakdown of glycogen. Glycolysis is this pathway down here. It produces 2 ATPs. When pyruvate is taken into the mitochondria, it will enter the Krebs cycle. So you see acetyl-CoA is the starting molecule for the Krebs cycle. And within the Krebs cycle itself, you can see that there's only one ATP produced per round of the Krebs cycle. You notice these NADHs and FADHs that are formed. They go to the electron transport chain where they can produce quite a high number of ATPs. So you get 3 ATPs per NADH and 2 ATPs per FADH. So the correct answer there is that you get most of your ATPs produced through the electron transport chain and a lower number through the Krebs cycle and glycolysis. Next question here, which enzyme would most likely increase during an endurance training program? So your best option here would be answer C, pyruvate dehydrogenase, and that's the best option because it's the only mitochondrial enzyme in this list. And it's your mitochondrial enzymes are the enzymes that will increase with endurance training. One of the major gases exhaled while breathing is carbon dioxide. So we exhale carbon dioxide at rest, and the amount of carbon dioxide that you exhale will increase if you're exercising. Question here is which metabolic pathway produces carbon dioxide? So the correct answer here is the Krebs cycle. So if you look at the Krebs cycle, you'll see that there's 2 points in the Krebs cycle where carbon dioxide is produced. Acetyl-CoA starts off as a 2-carbon molecule, so it essentially loses all of its carbons through formation of carbon dioxide. This carbon dioxide will then go into your blood and will be exhaled at the lungs. You'll notice that the pyruvate dehydrogenase reaction also produces carbon dioxide. So that's another possible answer to that question. And carbon dioxide is produced in the pyruvate dehydrogenase reaction when you convert pyruvate to acetyl-CoA, and then it's also formed in the Krebs cycle. So this is the carbon dioxide that you will exhale while you're exercising. Now, the main purpose of the electron transport chain is to make ATP to supply energy for muscle contraction. The question here is what substrates are required by the electron transport chain to accomplish this? So what has to go into the electron transport chain to get ATP as your output? And then the second question here is where do these substrates come from? So if you look at this diagram of the electron transport chain, you have ATP as your end product, and essentially the inputs to the electron transport chain include your NADH, your FADH, which recall can come from the Krebs cycle. NADH can also come from the pyruvate dehydrogenase reaction. And then you also have oxygen as your last acceptor of the electrons that flow down this chain, and they form water. And then you have ADP and inorganic phosphate that also feed into the electron transport chain to form ATP. So if I'm going to draw like a chemical reaction for the electron transport chain, and this chemical reaction isn't balanced or anything, but I'm just going to draw the inputs and the outputs. So you would have NADH that goes in. You'd have FADH that also goes in. And then you'd have ADP plus inorganic phosphate plus oxygen. Those would all go in as substrates for the electron transport chain. And they're eventually going to form, you're going to have NAD come out plus FAD. They lose their hydrogens. The hydrogens essentially go to oxygen as the last electron acceptor in that chain. So you get formation of H2O. And then you'll also have your ATP, which is kind of the most important part of that process. Now, as I mentioned, I haven't balanced off this equation, so I haven't drawn numbers in front of these. But these are the substrates that go into the electron transport chain. And then these are the products that will come out. The most important with regards to exercise is ATP, because then that can be used for muscle contraction. So the second part of this question was, where do these substrates come from? So the NADH and FADH, so far we've learned that these come from the Krebs cycle and the pyruvate dehydrogenase reaction. So essentially they come from a carbohydrate breakdown. So the carbohydrates that you take in through your diet eventually get broken down and allow formation of these metabolites. But you'll see later on in the course that they can also come from fat breakdown and protein breakdown. So essentially they come from all the foods that you eat. The ADP and inorganic phosphate. So the question here is, where did these come from that feed into the electron transport chain? These will come from anywhere in your muscle during exercise where ATP is expended. So this is a bit of a review. Recall that there's 3 sites in the muscle where ATP is expended. One is at myosin and actin, when you form your cross bridges for muscle contraction. The other sites are the sarcoplasmic reticulum, recall that you have to pump calcium back into the sarcoplasmic reticulum after contraction to allow relaxation to occur. And then the final site is going to be your sarcolemma. And that's going to be your sodium-potassium pump. Recall that after muscle contraction, you have to pump sodium out of the muscle fiber and you have to pump potassium back in. So it's the reverse of what happened during contraction. During contraction, you had sodium come in and potassium leave. And so during recovery, you have to reverse that process. So all of that will result in breakdown of ATP and formation of ADP and inorganic phosphate. So then that ADP and inorganic phosphate, if you're going to be resynthesizing ATP aerobically, those will have to feed into the electron transport chain in this equation, and then you get reformation of ATP. So it's like a kind of a circular process here. Breakdown of ATP at these sites, formation of ADP and inorganic phosphate, and then reformation of the ATP in the electron transport chain if your energy system that you're using is the aerobic energy system. And the last part of the electron transport chain is oxygen. That's the last substrate, and that will simply come from the air that you're breathing. So when you're exercising, your oxygen consumption increases, and the oxygen will eventually find its way to the electron transport chain to act as the last acceptor of the electrons in that chain to form water. So that answers that question about the substrates of the electron transport chain and where those substrates come from. Now this exercise is a bit of a review, and there's also a little bit of new material here. So it's matching these two columns, and this is a fairly difficult exercise that I'll walk you through. And keep in mind that on an exam, when I ask you questions about, I have a lot of enzymes listed on the right hand side here. You don't have to memorize the names of these enzymes. It's just important that you can recognize them if you see them on an exam, so on a sheet of paper. And by recognizing those enzymes, you should know their function and where they're located. So let's walk through this exercise here. I've got it reproduced on a piece of paper here, and I'm just going to put the numbers beside that correspond to each of these answers. So the first question here is catalyzes the exchange of hydrogen. And you can figure out the answer to this one by the names of these enzymes. So any enzyme that has dehydrogenase in its name is involved in the exchange of hydrogen. It catalyzes the exchange of hydrogen between molecules. So for this answer, you're looking for numbers 1, 2, 5, and 7. Okay, for the second one, catalyzes the breakdown of glycogen. Recall that at the start of glycogenolysis, you have this glycogen phosphorylase. You can just tell by the name of the enzyme what it does. Glycogen phosphorylase, it essentially breaks down your glycogen. Catalyzes conversion of pyruvate to acetyl-CoA. This is essentially the pyruvate dehydrogenase reaction. It's that important reaction that will take carbohydrate metabolism into the mitochondria to oxidize your carbohydrates or your glucose, essentially taking pyruvate in to get it converted to acetyl-CoA, and then that would enter the Krebs cycle. So your answer here is number 2. An intermediate in metabolism of carbohydrate, protein, and fat. Those can all be directed towards the Krebs cycle, and the start of the Krebs cycle is acetyl-CoA. So the answer here is number 6, acetyl-CoA. Catalyzes conversion of lactate to pyruvate. This is a mitochondrial enzyme. It is your lactate dehydrogenase H. So it takes the lactate from the cytoplasm and transports it into the mitochondria and gets that lactate converted to pyruvate, and then it can eventually enter the Krebs cycle. So your correct answer for this one is number 5. Catalyzes conversion of pyruvate to lactate. So this is the reverse reaction. This occurs in the cytoplasm, and this would be your lactate dehydrogenase M form. So your answer here is number 7. Enzyme of the Krebs cycle. So these are just markers of the Krebs cycle. There's many enzymes of the Krebs cycle, but these are the only 2 that you have to know or recognize, and that would be succinate dehydrogenase and citrate synthase. So you're looking at numbers 1 and 3 for that answer. An enzyme used as a marker of glycolysis. The only answer here, there's a number of answers here. This would be phosphofructokinase. You could also have lactate dehydrogenase M as an answer, and you could also have pyruvate dehydrogenase if you're considering that glycolysis can become aerobic. So we're looking at 2, 4, and 7 for your answers there. An enzyme of the electron transport chain. This is a new material, so this would be cytochrome oxidase. Number 9. Enzymes found in the mitochondria. We're looking at 1, 2, 3. And I'm going to continue this answer in the next presentation because I'm running out of time here.